Hello friends, this video on locomotion and movement part 7 is brought to you by examfear.com. No more fear from exam. Now the question is, when we talk about skeletal muscles, we see that they have a striated appearance. That is, they have striations. So here whatever you see, that is a particular muscle fiber. So this muscle fiber has the cytoplasm, it also has the multiple nuclei, and it also has striations, that is alternate dark and light bands. Now what gives this striated appearance? From where do this dark and light bands come into picture on these muscle fibers? Now there are myofibrils which are present in the sarcoplasm. So this is going to be interesting from here. So what are these myofibrils now? So it is again a new term. Now there is something very interesting here. Now these are filaments which are arranged parallelly in the sarcoplasm of the muscle fiber. So this was your muscle fiber, right? Now the striated appearance on the muscle fibers which you see, that is because of the presence of these thin filaments, and you will get to know that these filaments have alternate dark and light bands like this. Now they are extremely thin, but when many such filaments are joined together, so what will happen? Somewhere you have dark bands, somewhere you have light bands. So overall you will get a light and dark alternate appearance, and that is what is known as the striations. So these striations are due to the presence of these myofibrils, that is extremely thin filaments which are arranged parallelly in the sarcoplasm. Okay, so now here you can see the myofibrils. Each of these which are arranged parallelly, they are myofibrils. Now striations are seen on each myofibril, as I said. Now here everywhere, these dots which I am marking just to represent that they are the dark regions and the remaining portion is the light regions. So now on all these myofibrils you have alternate dark and light regions, and that is why you see them like this. Now this is how a myofibril actually looks like. I mean, this is how the entire arrangement looks like. So some portion is light, so this region is light region, again this region is dark region, again this region is light region and dark region, and so on. So alternate light and dark regions are present inside the myofibril. So this is the structure of a myofibril, one particular myofibril. So any one of these thin filament looks like this. Now the question is, what makes these striations to be present on each myofibril? What is there in the myofibril because of which there are alternate dark and light bands? Now these striations are due to the presence of two proteins, actin and myosin. So these are the two proteins which are present inside the myofibril. Now actin is the protein which gives rise to the lighter bands, and myosin gives rise to the dark bands. So here wherever you see light, that is due to the presence of actin. Wherever you see dark, that is due to the presence of myosin. So this actin and myosin proteins are present alternately on each myofibril, and that is why this gives a striated appearance on each myofibril. And since several myofibrils form the muscle fiber, that is why the muscle fiber also gets a striated appearance. Now the question is, how are these actin and myosin organized on each myofibril? Now as you can see here, if this is, now let me make a very rough diagram. Let us suppose this is your myofibril. Actually this is the structure of a detailed myofibril. Now let us suppose in that you have a dark band like this, then you have a light band. Again you have a dark band, you have a light band. Again you have a dark band, again you have a light band, and so on. Let us suppose this is how it is arranged, one dark, one light. So that means this is the portion where myosin is present, here actin is present, again myosin is present, actin is present, myosin, actin, and so on. So all these proteins are arranged just one after another, and they are arranged parallel to the longitudinal axis of the muscle fiber. Because see, this is the muscle fiber, right? This is the axis of the muscle fiber. So which would be the longitudinal axis, that is the perpendicular axis of the muscle fiber, would be this one, right? So these actin and myosin are organized parallel to the longitudinal axis of the muscle fiber, and they are organized alternately one after another. So they are arranged parallel to each other. So this is myosin, parallel to this is actin, again parallel to this is myosin, again parallel to this is actin, and they are also arranged parallelly to the longitudinal axis of the muscle fiber. So clear? So this arrangement has to be very, very clear, because only if you understand this, you will be able to understand the actual mechanism of muscle contraction. So this was your muscle fiber. Inside the muscle fiber, you have thin filaments called myofibrils. This is how a myofibril look like, and it is these striations on the myofibril is due to the presence of actin and myosin. Now let us talk about the two proteins, actin and myosin, in detail. Now actin and myosin are known as contractile proteins because these proteins are capable of causing muscle contraction. That is why they are called contractile proteins. So first we will talk about actin, and then we will take up myosin. So actin is the protein which is present in the light bands. So these are the light colored bands, and this is the place where actin is present. So this is the place where actin is present. Whereas myosin is present in the dark bands. So this is the region which is dark, and so myosin is present here. So actin forms the isotropic band, which is abbreviated as I band. Now what is the meaning of isotropic? The term isotropy means it means something where the properties are same throughout the region. So if you look at this light region, you see, now here in this picture, wherever you have the white colored lines, that represents the light region, that represents the protein actin, and wherever you have this dark blue colored line, that represents myosin. So if you look at the light bands, you see only white colored lines are present, right? So starting from here till here, everywhere you just see the white lines present. So that means the properties are same throughout. Properties in the sense, it is all made up of actin throughout. So the entire light band is made up of only one protein, that is actin. So that is why it is called isotropic, because in a way it is homogeneous. Whereas in case of myosin, it forms an isotropic band. That is because if you look at the dark band, you see the middle portion of the dark band is all dark. So in fact, till some places, so if you look here, it is only the dark blue, here also only dark blue. But beyond this, you have both dark blue as well as white. So in case of dark band, the middle portion of the dark band is all pure dark, that is, it is all pure myosin. So maybe only this much portion is pure myosin, but beyond this it is combination of myosin and actin. Again beyond this it is again combination of actin and myosin. So that means the distribution of myosin in the dark band is not uniform throughout. That is why it is called anisotropic. So here it is comparatively inhomogeneous when compared to the I band. So that is why the actin region is known as the isotropic band or the I band. So this is known as the I band, and this portion is known as the A band, that is the anisotropic band. Actin are thin filaments. Now these actin portion is comparatively thinner, that is also because structure wise they are thin filaments, whereas myosin structure wise are quite thick when compared to actin. In actin, there is an elastic fiber called Z line which is present at the center of each I band, which holds the thin filaments. Now here if you see, these are all thin filaments of actin. Now these thin filaments, you can see the white lines are drawn thinner when compared to the dark blue lines, just to show that actin filaments are thinner than myosin. Now if you see, this is the I band. At the center of the I band, you actually see a white line. So this line is nothing but the Z line. This line is called the Z line, and what does it do? It connects or it holds the thin filaments together. The filaments on this side and the filaments on this side, they are joined together by this Z line. Now, in a very similar way, there is a fibrous membrane called M line, which is present at the center of each A band, which holds the thick filaments. So here if you see this one, this structure which you see here, that is the M line. So I'm sorry about it, here by mistake A band is written as I band and thick filament is written as thin filaments. So this is the M line. So you got the structure, how the myofibril structure would be. It consists of filaments, because these proteins are also present in the form of filaments, thin structures. So the thinner ones are the actin and they form the light bands, which is isotropic, and the thicker ones are the myosin, which form the anisotropic band. So at the center of the isotropic band you have the Z line, and at the center of the anisotropic band you have the M line. So this is the basic structure of the contractile protein. Okay, now another important thing to note here is, now this protein, this entire myofibril is present throughout the length of the muscle fiber. So you will be having many Z lines and many M lines, right? Now the distance between two consecutive Z line, for example, this is one Z line and this is the next Z line. So the distance between these two, this distance, what is it known as? This distance is, or this region between two Z lines is defined as sarcomere. And this sarcomere is the functional unit of contraction. When we will study the process of contraction, we will see that as the sarcomere contracts, the muscle contracts and it causes the bones to move, thereby causing a movement. So the sarcomere is the basic functional unit. So in order to contract the muscle, you will have to contract the sarcomere. That means you will have to bring the Z lines closer to each other. That would be our aim when we talk about muscle contraction. Okay, so now, thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.